Hello students, this is the lesson video for lesson 4.01, the first lesson video for Unit 4 Mendelian Genetics, and we'll be going over the work of Gregor Mendel. So first let's go ahead and look at an overview of what we'll be going over today. So we'll be focusing on the work of Gregor Mendel, and we'll be seeing how he studied pea plants, in order to figure out different inheritance patterns, how he discovered the difference between dominant and recessive traits, and how he discovered what we now call genes. The goals for this lesson will be that you'll know Mendel's laws of segregation and independent assortment, that you'll be able to define what a gene is and what an allele is, that you'll be able to explain how the fundamental rules of inheritance began with his work and how they've been modified since his initial discovery. And you should be able to describe Mendel's experiments that led to the laws of segregation and independent assortment. Lastly, you should be able to compare and contrast the concepts of dominant versus recessive traits. Okay, so let's first look at what heredity is. Okay, so we have a really simple example. So we have this plant called the teosinte, and the teosinte is basically the primitive form of corn. And so the Native Americans had access to the teosinte, and eventually when they saw certain plants belonging to the teosinte family with desirable traits, they started to breed those with one another over and over again. And so what you see over time is we have progressively bigger and bigger plants, and that's how we get what we call today corn. And so this is an example of selective breeding where we have plants with desirable traits that are bred together. But it really shows you the power of heredity and the basics of heredity, because we know that if a certain organism has a certain trait, then it's able to pass that on to its offspring. Another example of selective breeding would be animals. So we have animals such as cattle and sheep and dogs that are all selectively bred in order to achieve desirable qualities such as maybe disease resistance, right, for farmers, or docile behavior so that they're able to be tame and kept in the home. So animal breeders really rely heavily on heredity. Early plant and animal breeders contributed important information to the study of heredity. In case you didn't know, a recent genetic analysis at the University of California confirmed that all dog breeds known today descended from one place, which is the wolf. Okay, so they all come from a common ancestors. And so although there are many different types of dogs, they all originated from that wolf. So many breeds arose hundreds of years ago as people intentionally mated, so that's kind of like that selective breeding, dogs to produce offspring with traits that would make them ideal companions, so they could be good working dogs or hunting dogs. There are other types of breeders, such as flower breeders. The German botanists experimented with crossbreeding different colored flowers so that he can come up with unique characteristics. And there are other breeders that breed chickens in order to have exotic characteristics such as this feather crown that you see right here. Okay, but really where our focus is going to be in this lesson is in Gregor Mendel, and he's known as the father of modern genetics, and he didn't deal with dogs or flowers, but he dealt with pea plants specifically in his garden. So he was an Austrian monk, and he did his experiments with plants, specifically pea plants. And unlike his predecessors, he used rigorous scientific methods. So he was very methodical and careful in how he did his experiments, and so he was able to come up with some really fascinating conclusions. So Mendel conducted his experiments with pea plants, which he grew in the garden of his monastery. The plants were really ideal because they were easy to grow, they reproduced really quickly, and they had some unique characteristics that were easy to observe. So he couldn't do these experiments with humans because, as you know, humans don't reproduce until later on in life, whereas plants can reproduce in a much quicker turnaround. Okay, so some characteristics that he observed of pea plants would include flower color, seed color, and also pea pod shape, and we'll take a look at some other ones as well. Mendel knew that each characteristic existed as one of two traits or form of character. For example, in the pea plants, if he was looking at the gene seed color, then he could see two different types of traits. He saw green seeds or yellow seeds. And so this was kind of the starting point of his scientific experiments. So again, one really key thing and characteristic about Mendel is that he had meticulous experimental design, always keeping close records, which allowed scientists to really respect his work and was convinced of his conclusions. Okay, so he studied pea plants, and again, one of the biggest reasons why he studied pea plants was because it had so many unique characteristics. You see here, they have different flower color, seed shape, pod color, pod shape, flower position, and plant height. All of these different characteristics, and then again, we have two different traits for each characteristic. You can either have purple flowers or white flowers. It can have a green pod color or a yellow pod color, etc. And so again, he applied these experimental methods to these pea plants, really studying how these characteristics were passed on from one generation to the next. And so what he first did was that he first grew groups of plants that he allowed only to self-pollinate. And so that means that they were making identical copies of themselves. And so the reason why he did this was so that he would make sure that each of these plants were true bred. So they're only producing offspring with the same character. So if the plant had purple flowers, they would always produce offspring with only purple flowers. And so he knew that he had a pure or true bred plant when he was starting. And that starting generation is what he called the pea generation or the parental generation. It's our starting point. Okay, and so he ensured that it was true bred or a purebred plant by making sure that they're constantly self-pollinating and producing consistently the same characteristics. Okay, so prior to Mendel's experiments, many scientists believed in the blending inheritance, which meant if you, for example, had a yellow parent, right, and a blue parent, if they were to mate, then they would make a green child or green offspring. Okay, so this is a very popular hypothesis among many scientists. However, Mendel did not come up with the same conclusion. Okay, so how he discovered this was he created an F1 generation. And F actually means filial, and that comes from the word meaning son. And so P generation was our first generation, right? And then after the P generation is going to make the F1 generation, right? Where you have all of the offspring. Okay, so Mendel took plants that were true breeding for green seeds and crossed them with plants that were true breeding for yellow seeds. So basically what he did was he took two plants that he knew were purebred, one that had green seeds, the other one with yellow seeds, and bred them together. And he was curious what color would their seeds be. And so how he did this was he dusted pollen from the yellow seed producing plants onto the flowers of green seed producing plants, and then did the opposite as well. To his surprise, the next generation of seeds did not come out as an intermediate shade of green and yellow. Instead, what it came out were all yellow. And so again, we started with a yellow seed and a green seed, but all of the offspring, 100% of the offspring were yellow. And so that was very interesting to him, and made him realize that blending inheritance was indeed incorrect. And so this first generation is what he called the F1 generation. So what he did next was he created an F2 generation. And the way that he did that was he allowed the F1 plants, remember all of the ones with yellow seeds, to self-pollinate. Remember, they all produced yellow seeds. The offspring of those seeds he called the F2 generation. So again, we started with the P generation, right, which was the true bred yellow and green seed. Then they produced the F1 generation. Then we self-pollinated, right? And we made the F2 generation. So this is when he had another surprise, which is when it wasn't half and half, it wasn't just yellow, but instead it produced a ratio of 3 yellow seeds for every 1 green seed. Okay, so it had a 3-to-1 ratio. And Mendel repeated the same cross for all the other characteristics. So instead of focusing on seed color, he might have focused on flower color or pod color, etc. And he found identical results. The F1 generation displayed only one trait, right? It only had one trait, whereas the F2 generations displayed 2 traits, but it wasn't half and half, but it was in a 3-to-1 ratio. So he had to come up, he had all the data, but he had to. So Mendel's findings were that he was able to show that inheritance was often a matter of some sort of individual factors that were being passed from the parent plant to the offspring plant. So he knew for sure that the theory of blending inheritance was not correct based on his findings. In each case, the traits he observed retained their distinct appearance. They did not blend or average out over generations. So based on his data, Mendel concluded that pea plants have distinct units of inheritance. Okay, so he called these factors, contributed by each parent to the offspring. He called these unit factors, but really was unclear about what exactly they were. So today, what we call these factors are genes. Okay, so each of those factors or characteristics that he was looking at is what we call genes, and they often come in twos, sometimes more than twos, but for this class, we're going to think of them in twos. And the different types within each factor are known as alleles. So Mendel used mathematical analysis to further determine that each plant must have 2 units of inheritance or 2 factors, one from each parent. So for example, if you had parent A had one green factor and one yellow factor, it could either give the green, right, to an offspring, or it can also give the yellow to the offspring as well. In addition, he proposed that parents have 2 units for each characteristic, and that when they form gametes, eggs, or sperm cells, each gamete receives only 1 unit per characteristic. So you personally have 2 units for every characteristic, whether it be eye color, hair texture, etc. But within your sperm or egg cells, your sex cells, you only have 1 characteristic. So that means if you were to have a child, you're only passing 1 of the 2 units of characteristic to them. So today we call these units alleles, and basically what alleles are different versions of the same gene. Okay, so for example, the gene we've really been looking at in this lesson is seed color, and the 2 different versions, or the 2 alleles, are green and yellow. So they belong to the same gene of seed color, but they're distinct in that because they're 2 different versions of that same gene, which is why we call that an allele. So essentially what an allele is a version of a gene. Okay, so we know that each individual has 2 alleles for each gene. You get one from your mom and one from your dad. If you were to have the same identical alleles for a trait, you get the same allele from your mom and your dad, then this is called homozygous. Homo meaning the same. Okay, an individual with two different alleles, so maybe you get one trait from your mom and a different trait from your dad, is known as heterozygous for a certain trait, and hetero means different. So remember, a zygote is the result of an egg and a sperm fusing. So can you explain the meaning of the prefixes in the terms homozygous and heterozygous? So remember, homo means same, so that means that the two alleles are the same or identical for a trait. Hetero means different, so if you're heterozygous, then that means that the zygote contains alleles that are different for a trait. Okay, and here's the picture that explains the same exact thing. So someone that's homozygous would have two of the same alleles. You see they have the allele for the yellow seed and allele for the yellow seed. But this individual here has a heterozygous genotype, which means that it's different. So they have one allele that's for the yellow and one allele for the green seed. The alleles for characteristics are usually assigned the same letter. However, uppercase means it's a dominant trait, and lowercase means it's for a recessive trait. Okay, so you see here, this is two uppercase letters, big Y, and so they both have the dominant. This person has the two dominant alleles, whereas this genotype here has two different ones, one of the dominant, one of the recessive. Okay, so one of the key findings of Mendel is that he actually came up with this law of independent assortments. And what that means is that alleles of different genes assort independently during gamete formation. So gamete formation is basically when you make sperm or egg cells. Okay, so this is when you're actually producing those sex cells within your body. In conducting his experiments, Mendel observed another pattern. The inheritance of one characteristic had no influence over the inheritance of another characteristic. So what that meant was the pea plants that had green seeds, that didn't mean that they had green pods. They could have green seeds and yellow pods, or yellow seeds and green pods, etc. So the scientists refer to this phenomenon as the law of independent assortments. So genes associated with different characteristics will assort independently of one another during gamete formation. Okay, so genes are not inherited together, but they're inherited independently. So the significance of Mendel's work was not recognized for many years. His findings and conclusions were really impressive because no one really even thought about what genes were or what DNA was or chromosomes. So the scientific community of the day just ignored his research because it was so different than what everyone else believed. Mendel communicated his results publicly, but scientists of his day did not recognize or understand how significant his work was. Decades later, scientists used microscopes, discovered chromosomes, and described what the process of meiosis is, which is basically when you create those sex cells. By 1900, 3 different scientists studying plant genetics recognized that Mendel's theories about how characteristics are transmitted could be explained in terms of chromosomes. And so when scientists were able to physically see what was happening inside cells, they saw these chromosomes, these individual units, right, in the nucleus. They're like, maybe Mendel's theories actually are valid, but it wasn't until they're able to actually physically see it themselves. So you see, Gregor Mendel had very, very impressive findings. He had very impressive experimental design on pea plants, and he discovered a lot of different things, different inheritance patterns that we talked about, the difference between dominant and recessive traits, how sometimes certain traits dominate and mask other traits. And he actually came up with a concept of genes and how specific factors or units of inheritance can be passed on from parent to offspring, and that was very revolutionary at his time. So here's some important vocabulary you need to know. Heredity, which is the passing of genetic information from parents to their offspring. We have characteristic, which is an identifiable or measurable feature of an organism, such as size, color, or texture. Remember, Mendel studied many different characteristics of pea plants. Allele, which is a specific form of a gene, and we have genes, which is a segment of DNA that directs the development of some inherited traits. So we can have the eye color gene, right? And some sample alleles would be like blue eyes, green eyes, brown eyes, etc. Right? We have different versions or alleles of a gene.